Okay, so welcome to our very first exam review session in Micro 2. So this week's exam is going to cover basically the first 3 weeks, so modules 1 through 3. So primarily mycology, parasitology, and enterobacteria ACA. So those are our primary 3 weeks that we are going to cover. As far as the exam itself, it is mostly multiple choice questions. I think there might be one one essay question. So I'm looking for the word. So one essay question on it. Otherwise, the rest is going to be multiple choice. And let me just peek here to remind myself, it is 70 points. So 70 points total. All right, now with that being said, I am going to make sure I share my screen so we can get started on the PowerPoints. I did send you a PowerPoint yesterday that we are going to look through. So you should have gotten that in your email. So this PowerPoint is just going to cover what your expectations should be for the test. So of course, the very first 2 weeks we did cover mycology and parasitology, and so those are huge with descriptions of what the organisms look like under the microscope and certain little other key characteristics that were mentioned with them. So what I'm going to do is pull up the picture PowerPoints for both mycology and parasite and go through the pictures with you. And as we go through the pictures, I'll be sure to mention key little things that you should remember for each one for the exam itself. Now, there are no pictures on the exam. If there is a picture, there might be one. If I remember correctly, maybe there's one picture, but it kind of goes, so it does go with the question. I can't remember if that one is on there or not, but if there is, there's one picture. Otherwise, the rest is pretty much no pictures, but because we're going to go through, you need to know these pictures anyway for the final lab practical. And as we go through, I'll make sure I point out the characteristics that you should know for the test. So you got these pictures in emails before, along with the answer keys, so you should already have these and been using them. And so we'll start with mycology first, since that was week 1. I'm just going to put it in the slideshow format here. Okay. So the very first one that comes to, again, as you're listening to this as a recording, pause it so you can test yourself before I say the answer. That is always helpful. So when you look at this, this is going to be Histoplasma capsulatum. Remember, histoplasma is a type of dimorphic fungi. Remember, there are 5 dimorphic fungi overall. And the dimorphic fungi are based on the premise that they can occur in both the yeast and mold phase. The yeast will occur at body temp, 35 to 37 degrees Celsius. And the mold phase occurs at room temperature, so right like around 25 to 30 degrees Celsius. I always remember that molds grow on my bread sitting on my counter at room temp. That's how I always kind of keep it straight for me. So again, there are 5 dimorphic. I'll identify them as we come. Histoplasma capsulatum is definitely one of the 5 dimorphic fungi. This guy, the key characteristic on him is what we call these tuberculated macroconidia. So they kind of look spiky, if you will. So that is the key characteristic for histoplasma. Next, we have Sporothrix shankai. Sporothrix shankai is another dimorphic fungi, so that's our second one we've seen. The key with the Sporothrix is it clusters at the tip of the conidiophore. So you can see where the arrow is pointing, how it shows it's clustering at the very tip of that conidiophore stem. I always think it looks kind of like a flower or a daisy or something. So Sporothrix shankai, besides it being a dimorphic fungi, and all the dimorphic fungi do cause systemic disease, they're the big bad boys that we're always worried about. Sporothrix also causes a subcutaneous infection, and this is the only one that we were worried about was subcutaneous. We had a bunch that caused skin cutaneous infections, but Sporothrix was the only one we identified that caused subcutaneous. And then it can lead into a systemic disease like the other dimorphic fungi. So remember, the nickname with Sporothrix shankai was rose gardener's disease. A lot of times people got this from gardening. They might prick themselves with a thorn on a rose. This one is our beaver tail or club shaped, and so this is Epidermophyton flucosum. So the key is that beaver tail or club shape, very round, broader ends there. So just as it sounds epidermophyton, it causes cutaneous infections, so skin infections. This is our little germ tube test. The germ tube test positively identifies Candida albicans. Our paintbrush is penicillium. Penicillium, again, is an opportunistic infection, said to look like a paintbrush. Here is another opportunistic infection, and this is fusarium. Fusarium is the sickle cell or canoe shape to it. This one's always so pretty. This is Aspergillus fumigatus. Aspergillus is another opportunistic type infection. Aspergillus fumigatus especially is kind of sometimes nicknamed farmer's lung. It's easily picked up out of the soil and again infects those immunocompromised patients. This is the one that also is said to have a foot cell at the base of the conidiophore. Another Aspergillus, this one is Aspergillus niger. The big thing here is the entire head is covered in sporulation or spores. Another paintbrush, so we got penicillium again. In this case, the key here is the pointed or what we call spindle-shaped ends. This is called Microsporum canis. So again, the macroconidia here has like, I think 5 to 8 septate, and then the key is that tapered or spindle-shaped end that it gets there. And so Microsporum canis, again, big cause of ringworm. Here is our mariner's wheel. So this is Paracoccidioides brasiliensis. So I always think that I'm sailing to Brazil. So Paracoccidioides brasiliensis, mariner's wheel. Again, it is a dimorphic fungi. So this is our third dimorphic fungi now that we've seen. Again, the club shape or beaver tail, so you should be thinking Epidermophyton flacosum. This is the thick-walled yeast, so you can see the really thick wall in the yeast stage here. This is another dimorphic fungi, and this is Blastomyces dermatitis. Blastomyces dermatitis, thick-walled yeast. And then our last dimorphic fungi is Coccidioides immitis. So in this case, the key is the rectangle or barrel shape that it can look like, as you can see by the arrows. So that is Coccidioides immitis, extremely infectious to lab workers. It's easily transmitted, so definitely has to be under hood. All fungus should be under hood, but this one definitely needs to be, it's very infectious. More germ tube for Candida albicans. Another Sporothrax shankai, where it clusters at the tips. Another Aspergillus niger. Again, Histoplasma capsulatum, the tuberculated macroconidia. Aspergillus fumigatus, so pretty. Again, the rectangle or barrel shape is Coccidioides immitis. All right, so hopefully that kind of helped trigger some mention of some key characteristics with each one. What I would do is make sure you go back to the original mycology lecture. Remember what all of your different cutaneous infections, remember we did talk about Tinea nigra, Black Piedra, and there's a bunch of trichophyton in there. We didn't see a lot of those pictures here, but we did mention some things extra that aren't on here. So just go back and relook at a couple things off of your original lecture from that day, from week 1, I should say. Okay, let's go into our parasite pictures now. So we're into week 2. We're going to do the same thing, pause it, so if you can try to identify it yourself without looking. All right, so the very first thing we come to is Dientamoeba fragilis. It is technically a flagellate, but it looks like an amoeba because it's kind of blobby-like. So this is Dientamoeba fragilis. The key on this is that it takes up it has 4 to 8 karyosome chunks inside the nucleus. So you see on the left side there's 2 nuclei, and you can see some little granule chunks. On the right side it's a little easier. You can definitely see 3 there. There's probably another one if you focus up and down. So again, that's the key in Dientamoeba, is those 4 karyosome kind of chunks inside the nucleus. Again, Dientamoeba fragilis is one of those that has no cyst stage that we are aware of. It's only the trophozoite stage that we know of so far. This is Fasciolopsis busky, or you can call it Fasciola hepatica. So this is one of those that looks identical. So it can be either one. All right, now you have Plasmodium, which is malaria. In this case, it's specifically Plasmodium falciparum. And I know that because in the ring stage, it's the only one that can have two little dots, karyosome dots, or chromatin dots, whatever you want to call it. So they look like little headphones, don't they? So this is, again, Plasmodium falciparum. The key on this is the ingested red cells, so this makes it Entamoeba histolytica. Entamoeba histolytica is our main bad boy pathogen of the amoebas. It causes amoebic dysentery and liver abscesses because it will basically travel via the bloodstream to get into the liver to cause disease there as well as the intestines. So the key is Entamoeba histolytica definitely ingests debris, bacteria, red cells, which you can see here. It will also have chromatoidal bodies, which you can't really see in this one, but we might see later. But again, ingested red cells are a very key sign of Entamoeba histolytica. And again, Entamoeba histolytica is one of the few that has very progressive movement. The rest of the amoebas, which aren't as clinically significant, tend to move kind of slow. This is the one that has very nice progressive movement. This is Babesia. So it is passed by a tick, very much found in the United States. The key is the tetrad or Maltese cross look that it can get. Here we have Wucheria bancrofti. Wucheria is the one that causes elephantiasis. So when it gets in, it likes to infect certain lymph nodes, typically lymph nodes on the lower trunk it likes to do, where it causes extreme swelling, something we call elephantiasis. Again, it is passed by a mosquito. Giardia, isn't he cute? The left side is the trophozoite, what we call the old man, because it looks like he's got eyes and is staring at you. So that nice pear-shaped look, he comes from contaminated water. The right side is his cyst stage. I always think it looks like a tulip inside there, like the petals with the leaves and stem. So that is Giardia. This is Iotamoeba bushlii. The key is the glycogen vacuole here. That is such a prominent feature of Iotamoeba bushlii that you should definitely know. Large glycogen vacuole. Again, this is Giardia. It's the Giardia cyst. So a nice little petal inside. In this case, this is Blastocystis hominis. The key is that it pushes the nuclei to the outer edges because it has a central body. So the nuclei are only found on the outer edges, and that's the key on Blastocystis hominis. Again, lots of people like to say it blasts the nuclei to the outer edges to remember that. This is Entamoeba hartmanni. It doesn't really cause anything clinically, but I always said the key that I knew is that it has this dark, dark ring to the nucleus, and that's how I always identified it as Entamoeba hartmanni. It is a small amoeba, and again, it has very slow non-movement. This is Chylomastix mesnelii, another flagellate. This is your Taenia egg. Again, both Taenia eggs look identical. Taenia solium and Taenia saginata look the same. You would actually have to see the worm itself to determine the difference. So remember, they have 4 suckers on the worm, but the difference is on the neck, I believe, of the worm. So Taenia solium, remember, is the pig tapeworm. Taenia saginata is the cow or beef tapeworm. Taenia solium is also responsible for causing cysticercosis, so it can go up into the brain. This is Entamoeba coli, so E. coli, and the key is how many nuclei it has. It can have up to 8 nuclei. Another Plasmodium, in this case it's the gametocyte stage, and this is Plasmodium falciparum. It is the only one that has that banana-shaped look in the gametocyte stage. This is your spiral-shaped trichinella. So trichinella is commonly found in muscle biopsies coming from raw pork or bear. You can skip, just ignore that. I threw that on a long time ago. I never have taken it off. All right, in this case, this is Diphyllobrothium latum. It is pointing to the lid, or what we call the operculum. I know when I first originally lectured through parasites, it was, like, I kind of laughed because it's like you have to trust me it's there. It takes a while to identify it, but there is a lid-like covering called the operculum here. On the other end, there is said to be a knob. It's not always that visible. But again, Diphyllobrothium latum coming from raw fish. Schistosomas. Schistosomas always have a spine. In this case, this is Schistosoma hematobium because it has the terminal or end spine. This can also be found in urine samples. Here is, again, our Entamoeba histolytica. It has the chromatoid body here visible along with the ingested red cell and then 2 nuclei. So Entamoeba histolytica. This is our packet of eggs called Dipylidium caninum. So again, it's a packet of eggs. Comes from fleas of dogs and stuff. Here is our one that is never passed as an egg. It's typically passed as larvae, and this is Strongyloides stercoralis. Skip that. I should take those out. All right. This would be Hymelolepis diminuta, no polar filaments. Remember, there's 2 Hymelolepis. There's Nana and diminuta. Nana has polar filaments. Diminuta has no polar filaments, so that's a key difference here. So no polar filaments, very clear in here. This is Trichomonas. The key is that undulating membrane on the side there. It is a flagellate, so it'll swim with flagella, but it has this undulating membrane. And again, we have Trichomonas tenax, which doesn't really cause anything significant, and that has no cyst stage. And then you have Trichomonas vaginalis, which is an STD, and you will see in the lab. Oh, my little sea monkey. This is Trypanosoma. So Trypanosoma sometimes can curve like that C shape. These guys are definitely found in the blood, as you can see from the picture. So there are 3 different Trypanosomas to remember. Trypanosoma cruzi is the one that causes Chagas disease, and it is passed by the reduvid or kissing bug, and that is found in the United States. And then you have your 2 Trypanosoma brucei that are found in Africa, and they are both passed by the tsetse fly. You have Trypanosoma brucei gambiens, which is Western sleeping sickness, and then you have Trypanosoma brucei rhodesiense, which is Eastern sleeping sickness. There is our Hymelolepis nana with the polar filaments coming off the ends on each side here. It's not very as clear in this picture. There's better pictures out there. This is our Trichuris trichura. He should be pretty identifiable. This is the one that is the whipworm. So he has these little plugs on the end that people always think they look like handles to a casserole dish. So Trichuris trichura. Another schistosoma. This is Schistosoma mansoni. It has the lateral or side spine. Again, Trichomonas with that undulating membrane on the side. This is Isospora belli, contaminated water. This is our I said the wrong one. Enterobius vermicularis. So Enterobius vermicularis is commonly known as pinworm. The key is it usually has a flattened edge on one side. It's not totally obvious, but it's flattened out slightly. And again, pinworm, a lot of times we just take tape, like Scotch tape, cellophane tape, whatever you want to call it, and then tape around the anal area looking for the eggs that the female worm likes to drop and lay at night there. It causes lots of anal itching. There is Giardia again. This is Clonorchis sinensis. It is called the Chinese liver. So it's Chinese liver parasite, Clonorchis. Balantidium coli, the only ciliate that we needed to remember. So it moves by cilia, and it's got two nuclei, a micronucleus, which is just round, and then this big macronucleus, which is kidney bean shape, and that's the key to it. It's a very large parasite. Leishmania. Leishmania love to be inside macrophages, as you can see here. There are 4 different types of Leishmania, so make sure you go back and review which each one causes and then how they're passed. So go back into your PowerPoint and look at that. So again, the only ones that aren't really in here, like Paragonymous stands out to me. I didn't put that in here. Paragonymous, remember, is the one that can also be found in the sputum because it is our lung parasite. The other thing that you should note when we originally lectured through parasites is there's several eggs that look identical to each other, so you wouldn't be able to tell under the microscope which one is which. So I'm just going to type them out. So yeah, Fasciolopsis buski and Fasciola hepatica look identical. You have the 2 Taenia, so Taenia saginata and Taenia solium. They look identical. And then the one I never really make you know the looks of it, but you should know that they look alike, is Metagonimus. And I think that's Yakagawa. I can't spell the last part. I'm just going to say Metagonimus and Heterophes. It's mentioned in the original parasite lecture. So all of these look identical, so we wouldn't be able to use them to tell each other apart, really. The other thing that I didn't mention everything. So there's lots of other stuff out there. So remember Onchocerca, volvulus, which is river blindness. And then there is Aloa Loa, right, which is the African eyeworm. Remember how each of these are passed? They have vectors that pass them. Like I said, look further into Leishmania. Let's see. Oh, I just lost it. Paragonymous, we mentioned. Stermani can also be found in sputum because it's a lung parasite. Oh, there's a whole thing that I just blanked on that I was going to tell you. Hang in there with me. Let me think through my PowerPoint if I can remember it. Let's see. I know there was something I'm forgetting to mention to you, and it's going to drive me crazy. You know what? I have to look it up because it's going to drive me nuts if I don't find it. I know. Hang on there with me. I'm sorry. But I want to make sure that you're well prepped. Again, you can always go back and re-listen to the lecture that we did together. I think we hit the amoeba as well. We hit the flagellates well. The hookworms look alike too. So remember, there's Ancylostoma duodenale and Necator americanus. These are our 2 hookworm eggs, and they look identical, so we just call it a hookworm. We didn't mention our roundworm, which is Ascaris lumbricoides. There was a picture of that to know eventually, but that's the roundworm. So that's what stood out to me. That's what I was thinking of that I didn't go through yet. So there's a few things. Let's see. Yeah, I think we mentioned everything else that I thought was good. But again, be responsible. Go back and relook through your PowerPoint lecture from parasitology, and that will help. So definitely add these onto your notes of things to know. And then, like I said, go back in there as well. All right. Okay, back to our original PowerPoint here. The 3rd week was all about Enterobacteriaceae, which are all prevalent, clinically significant bacteria that we see often. So remember, there are 5 characteristics to be able to be part of what we call the Enterobacteriaceae group. Entero means intestinal, so in some way these involve the intestines, whether it be the disease they're causing, diarrhea, or they're inhabitants of our intestines. But the 5 characteristics to be in the group is they're all gram-negative rods, they all ferment glucose, not lactose. Lactose is different because you'll think, well, Shigella and Salmonella are non-fermenters. which they are, but they're non-fermenters of lactose, which is what is in MacConkey agar. This is different. They all ferment glucose. They are all oxidase negative. They all reduce, or most of them reduce nitrates, nitrites, and most of them are catalase positive. So those are the 5 characteristics to belong in the Enterobacteriaceae group. Remember, you guys did a little bit on the IMVIC panel. The IMVIC panel is a great panel to differentiate amongst them all because there are so many. So again, it's indole, methyl red, voproskar, and citrate. This is coming off the lab PowerPoint. It was like the very last slide on there. So definitely know your IMVIC reactions here. The big one, especially, is E. coli is hugely indole positive. That's such a big characteristic for E. coli. The other thing to note is Proteus vulgaris is indole positive, whereas Proteus mirabilis is indole negative. Keep that in mind. There's other features to note here as well that I'll talk about in a second. Other reactions to note are the TSI tube reactions. So whether it's A over A, meaning yellow over yellow, or K, which is purple over yellow, whether it produces H2S. Remember, only so many produce H2S. Salmonella, Proteus, Citrobacter, and Edwardsella produce H2S. The rest really don't. So use that H2S production to your advantage when you are answering a question on TSI reactions. If it shows that it has H2S production, look for one of those 4, Salmonella, Proteus, Citrobacter, Edwardsella. That right away narrows it down. So definitely know these reactions for your test. Very much know these. You do not need to know the LIA reactions, just the TSI ones. So here are the key pathogens I want you to focus on for the exam itself. Remember, E. coli has a lot of different strains. You answered that in this week's study guide. So knowing enterotoxigenic E. coli is the traveler's diarrhea. Knowing enterohemorrhagic is their big bad boy one that goes with E. coli O157, and that it causes hemolytic uremic syndrome, HUS. Also know with E. coli O157, there's that specialized sorbitol MacConkey agar that we talked about. With salmonella, that's a good food poisoning one. There's a couple different types of salmonella. And again, salmonella is an H2S producer. Shigella is got different types as well. The big thing with Shigella is that it has a capsule. So a lot of times you have to heat it up to get through that capsule to test it. So keep that in mind, heating it up. Klebsiella pneumoniae just looks like mucus on a plate, very mucoid looking. Proteus, the key there was it swarms, and hopefully some of you got to see Proteus this week. It swarms on an agar plate. It smells nasty. Serratia, the big thing here is to remember that it is normally when things grow on MacConkey agar, they look either purple or clear, depending on if they're lactose or non-lactose fermenter. Serratia actually will get a red pigment on MacConkey agar, so that's kind of key. Yersinia, remember there's syn agar that makes it look like bullseye colonies. You have Yersinia enteroclitica, and then you have your plague, Yersinia pestis. And then there's a couple others here as well. So go back through and make note of anything key-wise, characteristic-wise, like I just went through, disease. Know the TSI and IMVIC reactions for those as well. All right, last but not least, I can't remember where we had this. If we had the HASIC group in Micro 1, or did we, I thought maybe we mentioned HASIC in Micro 1, but it pops back up now. So I'm going to have you, I'm going to write this out. There's a reason I'm writing it out. Make sure you really know this. But this is a group of bacteria that all cause slowly, I cannot spell, progressive endocarditis. So they are gram-negative rods that all cause slowly progressive endocarditis. So each letter stands for a bacteria. So we have Haemophilus, Acinetobacter. I'm not going to need you to know the second part. It just gets to be too much. Cardiobacter, Eikenella, and Kingella. So no HASIC. It's just those 5 together, all because they cause slowly progressive endocarditis. And then the last but not least, we just started getting into these different gram-neg tests. You did a whole biochemical worksheet on them. Maybe you did some of this testing in the lab. But anytime that we gram stain and we have gram-neg rod, the very first thing we do is an oxidase test. If that is oxidase negative, very commonly we do an indole test next because we're thinking it's an Enterobacteriaceae then. So we need to just know a tiny bit about these two. And I brought up my biochemical testing worksheet that you guys are doing. So here right away you see oxidase is the first one on here. Again, purple is usually the positive, negative is no color. There is a couple other oxidase tests out there, but one of the more common ones has purple as the positive color. Remember, in this oxidase test, you know, it's looking to see if you have this tetramethylphenylendamine dihydrochloride, my God, that's a long name, will oxidize to indophenol, which is a dark purple colored reaction, essentially. So again, all Enterobacteriaceae are oxidase negative. That's part of their characteristics. And then indole, definitely should remember that it's looking for the enzyme tryptophanase. So it's looking to see if it can degrade tryptophan into indole. Huge, that's what it's, the principle of it. Definitely remember that. And again, the positive is typically a red color. So, and that's part of that INVIC reaction. So make sure you take those into note as well, and you should be fine. All right, so that's it. Good luck, you guys. Let me know if you have any other questions. Otherwise, we'll talk soon, I'm sure.